हेलो फ्रेंड्स, आज हम इलाहाबाद विश्वविद्यालय के बीएससी की सिलेबस की डिस्कशन करेंगे। तो हम हिंदी और इंग्लिश दोनों वर्जन में टीच करेंगे आपको। तो यहाँ पर हम देखेंगे पेपर फर्स्ट सब्जेक्ट जूलॉजी बीएससी पार्ट 3 के लिए। जूलॉजी सब्जेक्ट के लिए पेपर फर्स्ट पहला पेपर है। पेपर फर्स्ट में पहला यूनिट है मॉलिक्यूलर बायोलॉजी। और तो देखें तो इसमें टॉपिक 1, 2, 3, 4 टॉपिक दिख रहा है। इन चारों को हम 1 बाय 1 बाद में डिस्कस करेंगे। तो यहाँ पर हम देखेंगे कि यूकैरियोटिक जीनोम ऑर्गेनाइजेशंस जस्ट वी आर डिस्कस हियर इन द मॉलिक्यूलर बायोलॉजी eukaryotic genome organization. Second one, unique and repetitive DNA consequence recombination. And third one, chromosome mapping in bacteria and virus. How to occur the mapping of the chromosome in the bacteria and in the virus. Just we are discuss in point number 3. Now fourth number, molecular basis of the gene regulation. How to gene regulate on the basis of the molecular. Just we are discussing the topic of fourth of the molecular biology. Then regulation in the prokaryotes inducible repressible system, that is IRS. Now just 4 topic discuss in the molecular biology. The topic 1 in the paper first of the zoology part 3rd. Just we are now write BSc third year. BSc 3rd year. Now second topic, immune system. Just we are know about that our body have a immune system, but immune system have a many types. But here now we discuss the topics cellular component of the immune system. Immune system का सेलुलर कंपोनेंट्स क्या क्या होते हैं और उनका सेलुलर स्ट्रक्चर क्या क्या है. व्हाट इज़ द सेलुलर स्ट्रक्चर ऑफ द immune system? What is the cellular components of the immune system? Just we are discussing the topic 1 of the immune system. Immunoglobulin. Immunoglobulin is the most important topic of the immune system. Just we are discuss topic number 2 of the immune system paper first. Now the major third topic in the immune system, the major histocompatibility complex. It means histone protein and histocompatibility complex, major histocompatibility complex in the immune system. What are doing the functions, what are the maximum histone proteins participate in this complex. Yeah, what is the component of this histocompatibility of the immune system? Just we are discussing the topic number 3 of the immune system of paper first in BSc part 3rd. Now the immune system and disease. What is the relation between the immune system and disease? What is the relation? That means the immune system protect from the disease. How to protect the disease? Immune system protect from the disease. Just we are discuss when the topic is more elaborate and given to you in detail form. So next topic के लिए तब तक आप इसके लिए स्क्रीनशॉट नाउ. जस्ट यू टेक द स्क्रीनशॉट नाउ. We discuss the next topic, somatic cell genetic. Now number 1 of the somatic cell topics, number 1 of the somatic cell genetics unit. heterokaryons and cell hybrids. How to do occur this cell hybrid, and what is the heterokaryons, or what is the homokaryons? Just we are discuss in the detail after the end of syllabus discussion. Just we are the discuss here the syllabus. Now second topic of this unit, gene regulation in the heterokaryons and somatic cell. Somatic cell and heterokaryons, how to regulate genetically, that means gene regulation in the heterokaryons and somatic cells. Now third topic is somatic hybridization studies in malignancy. Malignancy is the point when the general regulation deviate from its normal condition, that means malignancy. Malignancy, sometimes the deviation from the general or normal process, that means when any process is normal condition, but sometimes the condition deviate from its normal conditions, is known as the malignancy. Malignancy occupy in different conditions. Now very important topic, it is the very small topics, transposons. Transposons is a type of genes. How to regulate in gene or in cell, how to transfer in the cell, or how to develop this type of transposons gene. Now the somatic cell genetics unit is complete in the 4 topics, 1, 2, 3 and 4. Now genetic engineering unit, genetic engineering. In the genetic engineering have many topics. Now first one, scope of this genetic engineering. That means genetic engineering, how much elaborate, how much functionable, how much usable, that means scope. What is the level of the genetic engineering? As we discuss here, engineering, it means using the technology, while the genetic, that means the use of technology equal to the level of gene. That means combinedly we called the genetic engineering. When we are engineering in the DNA, RNA, transposons, a cellular level or genetic level is known as the genetic engineering. More scope when we are discussing the detail, given the detail account in this topic, just we are discuss the what is the profit of the genetic engineering and the scope of the genetic engineering. Now second topic, restrictions enzyme and their use in gene cloning. What is the restriction enzymes? Such type of enzymes whose the cut the gene in different positions. Many restriction enzymes cut the different positions of the gene, or such cutted gene develop a new gene, its mapping to the end of 3 and 5 prime and 5 prime to 3 prime. Now this is used the gene cloning. Now after the cutting the gene, just new gene develop is known as gene cloning. Genetic engineering have third topic, nucleotides sequence isolations. Just we are know about the nucleotides. Nucleotides 5. Nucleotides is 5. Adenine, guanine, thymine, cytosine and uracil. Some sign. Or just are setting the sequencing is AT, CG, and uracil, sometime replaced by thymine. It means sequencing isolation. When the process of isolation of the sequencing find the nucleotide sequencing isolation, what is the sequencing? What is the sequencing? Now in 5, purine base and pyrimidine base, purine and pyrimidine. But these 5 bases is known as collectively nucleotide. Adenine, thymine, guanine, cytosine and uracil collectively called nucleotide. Nucleotide occupy in DNA double helix. The positioning of AT, A, T, C, G, A, T. Here, uracil did not occupy and does not occupy. Why? The uracil occupy in R, N, A, while this is the DNA. So, you can take the screenshot. Analysis of mRNA and cDNA probes and their synthesis. Probe. What is the meaning of probe? Probe is a small part of the DNA and RNA which is taken as a for testing. Messenger RNA, cDNA. Analysis, how to form the cDNA and mRNA with the probes we are testing and analysis and their synthesis. So this is the topic next of the genetic engineering. Now second one, either next one, in vitro synthesis of recombinant DNA. What is the recombinant DNA? This is the word clear that recombinant. It means again combinant, any again combined DNA. Just we are the more detail given account after the teaching you the topics. And gene cloning techniques. What is the gene cloning technique? Two and more techniques to the gene cloning. Just we are the discuss here during the teaching you. Next one, non-coding, intervening, non-coding intervening sequence within the eukaryotic genes. What is the eukaryotic genes? Genes two types. Eukaryotic and prokaryotic. Prokaryotic is a primitive type. Nuclear membrane is not clear that in prokaryotes. Therefore, prokaryotic gene incipient are suspended in the prokaryote cell, while the eukaryotic gene is separated or enclosed by the nuclear membrane. Eukaryotic gene. Application of recombinant DNA. Recombinant DNA technology. What is the application? In the biology, in the medicine, in the industry and in the agriculture. Most important topic. Many important, many use of the recombinant DNA in the biology. Medicine is also. Industry, most important topic. Agriculture, mostly used in agriculture. Every hybrid fruits are occupied through this from the agriculture. Now next, recombinant DNA potential. Potential hazards of the recombinant DNA technology. Recombinant DNA technology, how much to damage, the any agriculture, industry, medicine and biology. This is the topic, potential hazards. That means disadvantage of the recombinant DNA technology. Just we are the discuss here. Genetic engineering of the paper first. Micro injecting gene into animal oocytes, egg and embryos. What happens when micro gene injecting into oocyte, eggs and embryos? We are the discuss what happened during the teaching you in the given detail. Explain what is the oocyte, what is the egg and what is the embryo. Every positions are the developing condition in embryology. Now, third year completed. Thank you.